title for today is Building Computers with Bacteriophage. The type of computer that we're keen about is not to compete directly with the one on the desk here. We're interested primarily in implementing computing in spaces where silicon-based computing isn't going to work. At least I don't see how it's going to work, such as inside every living cell in my body. Okay? So these computers are going to be slow. They're going to be very modest in their computational capacity. But that doesn't matter so much, right? If I could, for example, count cell division events, then I might only need to track a couple hundred cell division events. I might only need 8 bits as a register for combinatorial counter. And that would be a lot inside a biological replicating context. So that's a quick introduction to the computing part, but more to say on that. With bacteriophage, and I want to use the photograph shown on this opening slide to begin to talk about bacteriophage if you haven't heard anything about them. If you look at this photo, I could ask very naively, but hopefully, of the natural materials you see represented here, what would you expect to build a computer from? Right, and eventually, many folks will look to the sandy material and say, we could do something with that. We'd have to clean it up and refine it and take it forward. But we know how to do things with silicon. The bacteriophage are also shown in this photograph, but you can't see them directly. They're typically, in this picture, in the ocean. Bacteriophage are eaters of bacteria. They're bacterial viruses. And it looks like they are the most numerate, abundant, greatest in number, replicating natural systems on Earth. The ecologists now appreciate them well enough to estimate that Earth has about 10 to the 31st bacteriophage particles on the planet with us. That's a large number. What that translates to, for example, is in the surface waters of the ocean, you might have 1 billion bacteriophage particles in a liter of ocean water. These particles are typically small icosahedral capsids, about 60 nanometers across, packaging up genomes that might be 40,000 bases of DNA. I'll come back and explore some of those ideas in more detail. And so what I'd like you to leave this photograph with is the idea that were we to set out to build computers with bacteriophage, or perhaps from bacteriophage, I want you to think about the steps of refinement, the number of moves we'd have to make in converting the natural material, the found objects, into engineered objects. Much like when you think about engineering a silicon-based computer, you don't simply take that sand and shovel it into a new pile and say, there's my computer. Now maybe you can think of it that way, but I see a lot of computing objects that represent many more steps of refinement, processing, adding in of extrinsic information, shaping, and so on and so forth. So think of biology as a living material, a substrate, and as we begin to, through synthetic biology on top of genetic engineering, get better at working with these materials and reworking the materials and refining them, what new types of objects we can make. So I just want to leave you that meta notion. Don't think of biology as only things that pre-exist, but what happens when we grind it up and rework it. Okay, so these bacteriophage I've represented, they are eaters of bacteria. And so I wanted to show you a time-lapse movie of bacteria growing. Some of these bacteria have been infected with the bacterial virus. If the cells are infected with the bacterial virus, they'll turn green because we've added a gene onto the virus genome that makes the cells, when infected with the virus, turn green. And some cells will stay dark. Right? The cells are these blobs, these black blobs. These are about a micron long. These are E. coli. This is a time-lapse movie of a couple hours under an automated microscope. And the movie's going to run forward in time and backwards in time. So there was an infected cell up here. This is now running backwards in time, I hope. Right. Now it'll start over. Infected, and the cell bursts. And that's called lysis, and that's the release of probably 100 bacteriophage particles, which are too small to see under the light microscope, so the cell just looks like it goes away. Whereas the cell that was infected down here continues to grow and divide, and the virus, instead of blowing up the cell, has integrated its chromosome into the bacterial chromosome and gone dormant. Okay. So HIV is not dissimilar in that sense. It can infect and go dormant sometimes. This is the same virus in both the infected cells, so far as we know, identical genetically. And what this represents is in developmental biology, a cell fate selection process. You have to decide, are you going to blow up the cell or are you going to go dormant? And you can think of that as a 0-1 outcome. Within this natural system, there are molecular components that carry out the process. Right? Regulators of gene expression that sense the state of the cell and coordinate that information to help set the switch. One of the things that's interesting about this process, if I just zoom out and come back to planet Earth for a second, with 10 to the 31 phage on the planet, probably about 10% of them, being very conservative and underestimating, can make these types of decisions. So that's 10 to the 30th decision-making phage particles out there. And they're making this type of decision about once a day, once about 10 to the 5th seconds. So in other words, there's 10 to the 25th switching events like this happening every second on Earth. Right? They're not all cross-wired immediately, but they are cross-wired through the recycling of nutrients in the ecosystem. So that's a pretty significant switching capacity just running out there on the planet. Any questions about what you're seeing here, or bacteriophage as a quick intro primer? Yes, sir.
You say there's a decision made. What's the question?
So I'm putting a sort of persona into this. But in the context here, it's imagine you're the virus. You've just gotten into the cell. Is the best thing to do go dormant and hang out and maybe pop out later under different conditions? Or should you immediately reproduce, blow up the cell, and release your progeny? If you look at this movie carefully, again, these are genetic, so far as we can tell, these are genetically identical cells running in the same environment. Now, the microenvironment's a little bit different. This cell is alone. But if you look at the movie at the beginning, you'll see that the cell up here, which pops, is a bigger cell. Whereas the cell here that goes dormant starts out as a tinier cell when it was infected. The bacteria are naturally going through this cycle of growth, pinching off in the middle, and so you get a longer cell and then two smaller cells, and that just keeps repeating. The rod grows out at the poles and pinches. In this paper we published 5 years ago, we were curious to understand, under conditions like this, can we find any marker that is an indicator of what the outcome will be? And the long story short is we saw that bigger cells are much more likely to be popped. And we think that's because evolution selects for, when you're only going to blow up half the population, blow up the population that's the bigger cells so that you get a higher burst of progeny, right? And it just has to do with when the infection is with respect to the cell division cycle. So it's chance in that regard. Now, whether you want to call that a decision or not, but it's a two-state system, right? There's a switching event somewhere in there. Other questions? From this point forward, I'm going to talk about harvesting some of the gizmos out of these things and building data storage, logic amplifiers, and other stuff. We can come back if you want to explore phage biology. It's an interesting area of biology because it initiated a lot of molecular genetics and then sort of fell out of favor. But much more recently, as people have appreciated how many phage there are and that they're driving a recycling of the microbial systems in the ocean, and oh, by the way, those systems are doing photosynthesis, all of a sudden, the biologists are getting excited about phage yet again. Okay, so the next few slides are a reprise of a talk I gave here almost a year ago, where we had reported, not sure the paper had been published yet, but we had just gotten a 1-bit data register working in a chromosome. And the The interaction I had a year ago was incredibly helpful because we had been telling ourselves the wrong story about our work. We had been representing what we had been doing in terms of doing genetic data storage as engineering set-reset latches, and naively importing the architecture of maybe a cross-wired NOR gate, where you have feedback in electronics, leading to a bistable system. And in interacting with this audience and then writing things down for the paper afterwards, it became impossible to avoid the reality, which was we hadn't made a cross-wired NOR set-reset latch system. We made a two-state system without any feedback that was somehow functionally multi-state. So I want to just reprise that very quickly, and then I'll move into the logic amplifier work. This abstract representation here is meant to depict a fragment of a chromosome. All right? And what we're doing is we're engineering this region right here. The first thing you need to know is double-stranded DNA chemically has an orientation, which is defined by how the carbons are connected in the sugar in the backbone of the DNA, the ribose. This is called the 5' to 3' directionality of DNA. So what that means practically is double-stranded DNA ends up having an orientation which can be in one of two states. You can have a section of DNA, like shown here, that can be oriented to the right, or I could flip it and paste it back in and be oriented to the left. And from a chemical and physical perspective, that's it. Those are the dominant states that double-stranded DNA can occupy. So that we're going to immediately leverage as a pure digital data register at a single molecule level, right? So to unpack that a little bit more, this is a section of DNA. It can be as long as we want, so long as it's at least 25 bases of DNA long. We could probably push it up to 40 to 50,000 bases long. In the work we're doing, it's about 50 bases of DNA long. So the region we're flipping is 50 different letters. Okay? The reason we're going to choose to flip a sequence of DNA that has so many letters is so that not only do we have a 0-1 register, but the thing we're flipping has some genetic activity. It tells the cell to do something. And in the simplest case, what we're going to flip is going to be called a promoter. This is a region on the DNA that recruits an enzyme called the RNA polymerase to bind the DNA and initiate the reading out of DNA. Right? Initiation of transcription. So if the promoter is pointing to the left, we'll read out the genes to the left. If the element with its promoter is pointing to the right, we'll read out the genes to the right. Now these triangles, okay, on the outside of this flipping element, those are stolen from bacteriophage. These are the genetic signals that bacteriophage use to do something a little bit different. When a phage infects a cell, if it decides to go dormant, it has to solve the problem of integrating its genome into the host genome. And it uses an enzyme called an integrase to do that process. The integrase is an enzyme that binds specific sequences on DNA, these triangles, and does that push-paste reaction. By changing how these sites are oriented, as discovered in the early 1980s, instead of an insertion, you can get a flipping. All right, so that's what we're reusing. So just to repeat this a little bit, we've got a stretch of DNA, call it 50 bases long. If we flip it into this orientation, we can read out a gene to the left, call that a 1. Maybe that gene makes the cells red. But it could be any gene, right? It could be the gene that controls flipping of another register, for example. If we flip it back, that gene turns off, another gene turns on, something else turns on. I hadn't fully appreciated at the start of the project how nice this property of DNA is. It exists in one of two states in terms of its orientation based on the backbone chemistry. That's a pure single molecule digital register. Okay, so we call this, colloquially, we call this thing the Flippy. And I had mentioned there's this enzyme from the phage called the integrase, which does this magical thing of typically in nature inserting, but by recrafting the sites, we can get this flipping reaction. So if we can just create a pulse of this enzyme in the cell under our control, then we can drive that 0 to 1 transition. Any questions?
DNA sort of was, the chemistry of attaching to its neighbor was sort of one directional. Where did this flipping idea come from? Might have date?
Repeat the last part of your question, please.
I don't understand. I thought DNA sort of had a fundamental orientation.
Right. So this is the single-stranded DNA does, right? It's sort of 5' to, it is 5' to 3' along one strand. But when you have double-stranded DNA, the complementary strand has opposite orientation. So it's 5' to 3' along the top strand, and then running the other direction, 5' to 3' along the bottom. That allows you to do the flip. Yeah, so I should have emphasized double-stranded DNA. Yeah.
So can flip it. How do you choose which flip you get?
Haven't shown the reset.
Okay.
A couple other things to note. If I have one integrase, I'm showing it as a cartoon, but this is a physical object. Right? The way it works is, and this is not perfectly understood, the way it works is the integrase binds a specific sequence on the DNA. Right? So the specificity of the data register is determined by the protein-DNA interaction, such that if I want to have a second register and I would like it to operate independently, I'm going to need to harvest and repurpose a second integrase with different DNA binding specificity.
The flipping, how often does it screw up or not proceed according to plan?
Spontaneous switching rates are less than 1 in 10 to the 12th cell division events. Let me show you some more data. They're really good. I mean, shockingly clean for biology. But let's come back to that. I just want to emphasize, if you want 8 bits, you need 8 integrases at least. Right? If you're going up to toggles, you might need 16. Right? Right now in my laboratory in Y2E2, the team has 4 integrases sort of under control that recognize different DNA binding sequences. We know that there's a ton of phage out there, and over the last year, just by starting to publish this work, we've created a new demand or market for people harvesting integrases that didn't exist before. And there's a really neat program run out of University of Pittsburgh, sponsored by Howard Hughes Medical, that's called Phage Hunters, where high school students basically collect phage and crowdsource the sequencing and annotation. So I'm hopeful that they're going to be part of the solution to getting more gizmos. Some integrases that you can find are not unidirectional, meaning they will flip, but then they'll spontaneously backflip, okay, or reset, and that gets to the reliability issue. Jerome Bonnet, who I'll introduce you to in a moment via photograph, selected a type of integrase called a serine integrase that is really nicely unidirectional. Okay? Moreover, the enzymes we're down selecting to don't require any host cell cofactors. So all the work I'm showing today is done in E. coli, but the phage molecules we're using have been shown to work in plants and mammals and so on. So the biochemistry we believe to be portable across kingdoms of life. The reset operation takes the same enzyme and adds to it what's called a recombination directionality factor, or historically has been called the excisionase. The excisionase, as you can imagine, in nature is the enzyme that, when present along with the integrase, causes the integrated virus genome to excise or pop out, leading to blowing up the cell. So if I take these 2 proteins and I express them together, that changes the directionality of the reaction, and I can reset the DNA data register to the exact sequence I started from, completing a cycle. Okay? In the abstract? So this was basically the win slide from a year ago. What we're showing here is the greenness of cells on the y-axis. This is data collected from a cytometer, where basically flowing cells and getting a very quick microscopic snapshot and measuring their greenness. So there's a distribution of single cells. So they're starting out in the 1 state. That's what a low in green means. We give them a reset pulse. They become green. And when we remove the reset pulse, the cells maintain their greenness. They're storing the green state. If we then give them a set pulse, they lose their greenness. They become red, which I'm not showing. And they hold that state. Now, there is dispersion around the greenness or lack of greenness, and we believe that to be spontaneous, right? Coming from who knows what. People would say noise in gene expression or I don't know what. This is a log-wise scale, so the means are probably shifting by 50-fold in terms of expressed protein intensity. And that's because through this digital transition in the orientation of the promoter, we can go from a lot of expression of the left gene and little of the right and vice versa. So we get a pretty nice swing. And we can repeat this. So what was new a year ago was showing for the first time that you could close the cycle and then repeat the cycle. People had previously shown just setting, one-time setting, not rewriting. These input pulses are quite long. You can see it says 10 cell doublings. So that might be, in this experiment, 12 hours. A 12-hour input pulse. Okay? So if you wanted to make a cell division cycle counter, a 10-cell doubling switching requirement, not so good. So we've been pretty keen to push the speed. And this is what it looks like if you cut the pulse size down to 2.5 doublings, okay, which probably isn't fast enough yet. We have seen in movies now that we can get switching with a 15-minute input pulse in cells that are doubling every hour. So we think we can get within the cell division cycle window in terms of timing, which is key because cell division cycle counting is a big application area. Or underlies many application areas, I should say. What I had spent a lot of time talking about last year was how difficult it was to get this project to work. That it took us 750 attempts to do different DNA designs, to precisely control the expression levels of the proteins, to get no switching in the absence of inputs, reliable and complete switching in the presence of inputs, and no cross-contamination between the set and reset reaction, because they both rely on the integrase. If the excisionase shows up in the wrong moment, it'll corrupt a set and so on. So I want to summarize a couple properties about this, which really came out of last year's talk. This isn't a formally bistable system, so far as I can tell. There's no feedback. It's a non-volatile data register. You express the genes, the data register sets or resets, and then you get rid of gene expression, and then the DNA just sits there, and it gets copied if the DNA gets copied as the cell reproduces. But you don't have to keep expressing genes, right? Prior data storage systems in biology have relied on cross-regulating transcription factors, like protein A is turning off protein B, or protein B is turning off protein A, and that means you always have to be expressing proteins in order to maintain the state of your data register. But this is a non-volatile data register with respect to requiring gene expression. We don't require it once you set the register. And it doesn't seem to require feedback to hold state. This is very interesting to us. It allowed us, for example, to grow out the data registers for 100 generations and see if they'd be selected against. Would the mutations cause them to be erased from the population? They're evolutionarily rock solid out to 100 generations. They don't seem to spontaneously switch. We think they work because we have really nice analog to digital converters. We can take a continuously varying gene expression signal from no gene expression to high gene expression and everything in between, and at some threshold, flip the DNA or don't flip the DNA. We can do that in both directions. And that's what really came out of our realization that we hadn't made a conventional cross-wired NOR SR latch. Which then begged the question, what else can you make with A to D? And so that's what we had in our minds last May. Now, the people here, Tone and Jerome, left and right respectively, are the folks who have done this work and the work I'm about to discuss. And in addition to thinking about what else can you make with A to D converters, Jerome, having just spent 3.5 years getting a 1-bit data register, was pretty excited to do something else. And he was keen to try and take genetic data into medical diagnostics. Could you, for example, instrument microbes as part of an early detection system for diabetes, where biomarkers coming out in your urine, if you could integrate combinations of biomarker signals, might give you a upstream in time sense of how your disease state is emerging or progressing. And he pretty quickly found himself talking with people who are scouring for biomarkers in the urine and realizing that there's not one magic biomarker, but you have to do integration of multiple biomarkers. And so he started to think about, how could I do an AND operation? How could I do a higher order Boolean operation? So we took those two things together, and I'll sort of tell you the story backwards in a way, came up with a new way of thinking about implementing genetic logic. And it's based on a switch of the type you can now hopefully think about. We call the switch a transcriptor because we think it has similarities to a 3-terminal architecture used in the transistor. And it's regulating something akin to current, but not an electrical current, a transcriptional or gene expression current. So from these triangles, you should recognize that this is going to be these bacteriophage integrase sites targeting flipping. But now we're going to flip something different. We're going to flip this T. The T represents an element on DNA called a transcription terminator. Okay? The terminator is an element that stops RNA polymerase. Once it's going along and reading out DNA, it stops it, says no more gene expression past this point. All right? And these Ts are a particular type of T. They're asymmetric. So what that means is they block transcription only in one direction on the DNA. So if polymerase coming from this direction would stop, polymerase coming along the other strand would pass through. I don't know of a natural example. Somebody might send it to me. I'd be really grateful for this. I don't know of any natural example of people of nature having evolved the flipping of terminators. Right? There's many natural examples of flipping of promoters, but we haven't come across this type of architecture in a natural system yet. So the way this works is we have this enzyme, RNA polymerase, which reads out DNA. You think of it as a transcriptional current. And if the terminator's in this orientation, no pass-through. If I have a control signal come in, make an integrase, and flip. Now, because it's an asymmetric terminator, in this orientation, the polymerase can go all the way through. So if my input signal is high, I have a lot of input signal, I have a lot of output signal, just pure pass-through of the transcriptional current. If I flip it back to its original state, output's low. Right, so the input's going to be the transcriptional current on the left, the output's going to be the transcriptional current on the right, and the control signal is going to be the thing that changes the state of the logic element, right, or the switch in this case. Any questions about this? Yes?
Have you observed any changes in the overall behavior of the cell itself from flipping its terminators?
No, these are exogenous elements we're bringing in. Right, so we're not yet using this to rewire the transcriptional program, if you will, of a natural cell. We're thinking about that and setting that up, but right now we're just bringing in our whole new package that's insulated from the transcriptional architecture of the cell to begin with.
So that sort of change has not happened accidentally either?
No. No. Other questions at this sort of basic switch level? Okay, so if I have two integrases that recognize different sites, and I connect them in series along a piece of DNA, this is going to get me an AND gate. All right. So in this configuration of the logic elements, either of the terminators would block. If I flip via control signal 1 the first terminator, and I flip via control signal 2 the second terminator, I get input to output pass through. If the original orientation of the terminators is reversed, now they're allowing pass-through at the outset. Right? If neither control signal shows up, I get high output. But if either or both flip, then I block. This gets me a NOR. So just by a simple reconfiguration of the DNA in the logic element, changing the original state of the terminator, I go from an AND to a NOR. Here's where it gets interesting compared to conventional electronics, I think. So this is what an XOR looks like. The output is high only if the inputs are different. In this system, in this logic element, we've gone back to just having a single terminator, but now we're, which is flanked by flipping sites, but now that whole package is flanked by additional flipping sites. Right? So in this 0-0 input configuration, output's low. If the first control signal is high, then the terminator's flipped and the output's high. But if the second control signal comes in, the whole package flips again. So I'm able to integrate multiple control signals at a single point on my DNA wire. It'd be like having a transistor, and I don't know if can this be done or not. Can I have a transistor and have it switch, the same transistor switched by multiple signals coming into the wire at the same point? That's what I think we're doing here in the DNA. And as a result of that architectural feature, we can implement an XOR in a single layer, right? Whereas I think in TTL or something like that, if you wanted to implement XOR, you'd have to layer some gates. And I have to admit, I'm not the electrical engineer, right? So somebody who's expert in that should be correcting me if this is off base. But this was pretty exciting for us, and it was in part pretty exciting because most of the genetic engineering work around logic gates had been following the architectural models established in electrical engineering. So people had implemented XOR gates, but they had done so by layering many simpler gates, if you will.
If you want input from electrical engineers, the transistor analogy isn't so strong. And you slip into actually calling it a transistor when you initially introduce it as a transcriptor. There's gain there, and it's not a binary thing. And you show bipolar NPN transistors, and they don't work the way you described. I mean, there's a loose analogy, but I'm worried about people starting to actually take it literally.
Yep, fair enough. I think there are important differences.
Relay logic is more like what this is.
Yeah.
So they built transistor computers in the 50s, where you can latch a relay, you give it a signal, and it stays in one state, and then you can unlatch it later by putting a current on the other side. Transistor, like you showed, has a gain where if you use the term current, RNA will be coming in one terminal or some molecule, and there'd be a proportionality of the output signal.
Let's get to that. I mean, I think you're making a good point, right? The analogies sort of get a conversation going. They could derail a conversation too. I'll show you what the transfer functions look like. Yeah, and let's keep talking. But I do want to represent one thing. Because we live in a world where electronics has dominated, right, or dominates a lot of thinking, the genetic engineering community had actually implemented the types of architectures depicted at the bottom, which were hugely expensive to implement in biology, and just didn't work very well. And so it was exciting to sort of recognize biology as its own material, right, and come up with an architecture that's just nice. Sorry?
Have you built one of these?
Oh yeah. XNOR just flipped the original state. I won't belabor this. These are our first generation logic elements. They're permanent logic elements, as I'm showing you. Or so they write once, and they can receive asynchronous control signals and integrate them. It's simple to make rewritable elements that get pushed and then relax spontaneously. So we can go into that. But in any case, let me zoom out. So at an abstract level, we've got an input signal, which is a transcription signal coming in. We've got an output signal, which is a transcription signal going out. And we've got control signals, which are transcription signals as well, transcriptional current as well, going into the recombination controller, which means they're making integrases when they come in. And we slot into this logic elements. The types of logic elements I've been showing you are these two input logic elements. These are write once elements. Okay. So we built all these to start. And we started working on this in June or July of last year. This is what we would predict the logic elements would do in terms of their output state in response to control signal 1 and control signal 2, based on models showing what the propensity is for flipping DNA at different levels of the enzymes. And when we built these and put them into a population of cells and just measured the bulk properties of the cells, this was the initial data we got back. Now this is a log scale here. No, excuse me, it's a linear scale. And so, for example, you could see that the NAND gate isn't completely symmetric, right, across all the output states. It's about a twofold difference here to here. And the OR gate as well is a little bit different. When we looked at the distributions of single cells and what the individual output levels, as measured indirectly by a fluorescent protein, we can't measure the flow of the RNA polymerase, this transcriptional current, if you'll permit me that. We can't measure that directly yet. But when we look at the output indirectly through the fluorescent protein levels, and this is now on a log-Y scale from left to right, you can see the changes. You can see that you don't have everything being exactly matched. But nevertheless, across this whole family of logic elements, we can identify a threshold that can discretize the output between low and high. And that's what that red line represents. And then score our false switching rates, if you will. What type 1 and type 2 errors on the switches. This is a detailed example of one of the switching, both switching states for the XOR. What I'm showing on the y-axis is an indirect representation of the signal. Again, it's a fluorescent protein serving as a surrogate for the rate of transcription. The blue contours are the level of the control signal that's driving the integrase. And the red contours are indicative of the output signal. Okay? And so in this case, the XOR gate is getting one of its inputs, and that input is going up, as you can see in blue. And across this change in the input, we get a snapping up of the output. So we believe that the gate's switching, and the output goes from low to high. The terminator has flipped. Here's with the different control signal, the second control signal for the same logic element. This is now driving a different integrase with its own peculiar biochemistry, slightly different sensitivity levels. And we get switching across this region, but with some cells not completely switching, right? And you sort of have to look across a bigger range to see the switching. This was pretty exciting for us because the change in the output's bigger than the change in the control signal, is what it looks like. That you can wiggle the control signal a little bit and get a pretty significant change in the output. Again, this is on a log y axis, right? So we might be going from a arbitrary value of 20 here to 50, whereas the output goes from 50 to almost 3,000. So that's a pretty big difference in terms of the relationship between the change in the output signal and the change in the control signal required to switch. The XOR gate is our best gate right now in terms of clean, discrete amplification, or however you want to describe it. And if you look at the paper that just came out this month, last month, you can see our analysis of switching failure rates, if you will, for all the gates that we initially developed. Any questions about this, or do you want to return to metaphor or not?
Question, what do you, if you're relating to electronics and computers, what do you make your clock speed out to be for computation?
Millihertz? I don't, what's the right units to use for clock speed? The switching times, the minimum switching times in terms of the control pulses are 15 minutes right now.
Okay.
And the way I bring that back to the biology is if you think about bacteriophage infection, which is where we're harvesting these parts from, that decision has to be made and implemented on the timescale of tens of minutes. So I'd be surprised, just physically, if we could drive things much faster without creating new selective pressures to push the enzymes. I don't know what the limit is. So we have 4 integrases. We could probably barely just implement then two input gates that operated independently, right? Or a higher order gate that integrated signals. We come back to this if you want. I wanted to share a third result over last year. This is work done by Monica Ortiz, a graduate student in BioE here. And she's developed this platform for cell-cell communication that is, again, based on reusing bacteriophage. And what she basically has done is parasitized the virus such that the cell engineered with her system is infected with a virus, but instead of the virus packaging up its genome, the virus will package up any piece of DNA that she targets with her packaging signal. That chimeric virus particle, or transvestite virus particle, if you will be secreted from the cell. We're using a bacteriophage that does not blow up the cell in this case. So the cell survives infection and can replicate and just continues to broadcast these virus particles containing Monica's messages. Okay? These virus particles then go out into the environment and they infect other cells, but they're infecting it with her engineered messages. All right? The significance of this is it's the first time in bioengineering where the channel for communication has been decoupled from the message being transmitted. Previous work on engineering cell-cell communication has relied on sending and receiving small molecule sugars or peptides from bacterial quorum sensing systems, if you happen to be familiar with that. And in that context, the message being transmitted is the molecule itself. Right? So schematically, you should recognize the top of this lower figure. That gets implemented by having an infected cell which can package these arbitrary engineered DNA messages. The messaging particles get released into the environment and they transduce other cells. You can cause this system to, in addition to transmitting the message, also transmit infectious virus, which can turn a population of receiver cells, if you want, into rebroadcasters. Okay? So you have that option. So I'll just give you okay, let me say one thing in setting this picture up. The weakness of transmitting messages as DNA molecules as opposed to sugars or peptides is the DNA molecules tend to be physically larger. Right? The virus particle in this case is a interesting shape. It's actually a rod, maybe 600 nanometers long and just under 10 nanometers across. Right? So its spontaneous diffusion rate is quite limited, maybe half a millimeter in 24 to 48 hours in a jello-like substrate, as opposed to these small molecule sugars, which go 2 centimeters in the same period of time, just spontaneously. So one of the things Monica did that was pretty neat was to overcome the limited broadcasting range in these bacteriophage messaging systems by coupling the packaging of the message and the secretion of the particle with active cellular chemotaxis. So what's shown here is the result of such an experiment where she's on a 10 centimeter diameter petri dish plated cells that are initially red here and initially green here. And over a period of time, I think it's about 24 hours, they replicate but also swim into the fresh media, such that at the end of the growth experiment, the green cells have grown from west to east and vice versa with the red, and they meet in the middle, and they've, at this point, have depleted the plate of nutrients, primarily. But this is a pretty big gap for a cell that's a micron. This is a 7 centimeter gap being, transited in a day or so. If you look at what's happening at this boundary, I honestly can't remember right now if the green cells are broadcasting or receiving, or vice versa. But suffice it to say, broadcasting and receipt is happening, and some cells turn yellow. Okay? In the limited window of time in this experiment, where not much can happen because as soon as they collide, they stop growing and everything shuts down, we're seeing about 30% of the cells getting receipt of message. Okay? If we do this experiment in a liquid culture where everything's mixed up, the spontaneous receipt of message rate, meaning what I mean by spontaneous is you don't do a genetic selection to see if the message got there. If you do a genetic selection, say only the cells that got the message can survive. If I do that, I'll get everything growing that receives the message. So if you want 100% receipt of your message, make your message essential, everything else dies. But in the absence of selection, we get about 30% transmission on this semi-solid media. In the absence of selection in liquid, we get about 93, 95% transmission without doing an evolutionary selection. Okay. So you can communicate these logic gates, right? And that's shown here. This is an AND gate being communicated. What's neat about this is we can communicate because they're permanent write once gates, we could communicate the intermediate state of the logic element. Right? So we could have the logic element be transmitted where no input signals have come in, no control signals have come in, excuse me, if one or the other or both control signals. So the messages being broadcast can encode the intermediate logic state, for the permanent gates at least. And that hadn't been shown before. Okay, so a reprise of a digital data register, an introduction to amplifying logic elements, permanent elements here. I can go into details around the rewritable and how to compose higher order inputs. 15-minute input pulses required, best case scenario. We've got 4 integrases. Right? So the scaling up is going to be limited by harvesting of the biochemistry. We're very optimistic about the natural biochemistry and the diversity out there. I should mention that there's if you just think through this, because these enzymes are the enzymes responsible for integration of the virus into the host genome, they're under selection to not choose the same DNA sites to integrate into. So we expect we're going to be able to get molecular recognition diversity in terms of the natural integrases. We won't have to engineer the integrases to bind different DNA, although people are trying to do that now. Okay. In the context of engineering biology, this is our simplistic representation of the core of the engineering cycle. How long does it take us to design something? How long does it take us to build it? How long does it take us to test it? And how many times do we have to go around this cycle to get something working? With the data register work I started with, that took us 750 design attempts, loops around this cycle, and thus 3.5 years. However, coming from this course a year ago and recognizing what we think of our work is now A to D converters, we abstracted that, reused them. When we implemented our logic amplifiers, every design worked the first time. And I've simply never had that experience in engineering biology, only needing to go around this cycle once. I'm still trying to figure out, I like it, but I'm still trying to figure out what to make of it. There's background work, just for context, trying to improve this cycle, and it mostly collapsed over the last decade into 3 areas of fundamental engineering work, using DNA synthesis to get better at decoupling design from fab and scaling that up, standardizing measurement and componentry in support of functional composition, such that you can make reusable parts like the A to D converters, and then trying to abstract things up from the biochemistry into A to D, into latches, into switches, into whoever knows what comes next. Right. And so I just want to tease you with the fact that in bioengineering, one of the fundamentally important areas of work is this fundamental cycle improvement work. Faster, more times in parallel, more smartly, I don't care. But all improvements are good. And what I can represent today that I couldn't represent a year ago is after a decade of work across many labs, we're hitting some milestones that are pretty interesting. You know, we can now do forward compile of, this is going to sound incredibly modest, but to genetic engineering it's a big deal. We can now do forward compile of 10 gene systems at a time and get those genes to be expressed to precise levels inside the cell with very, very high reliability. That outstrips our design capacity right now. Very few people know how to specify the design of a system where they tell you up front that they care about the exact concentrations of the proteins over time that they want, because we just don't know enough to design, or people haven't thought through that completely. So that's an aside, but an important transition underway. So happy to come back to, well, let's see, it's 5:06. So we go to 5:30 or 5:15 or when? Are we done?
30. Or tomorrow morning at 8.
Well,
It's you're right,
9:30. But 5:30, right? But what? So okay. So at this point, we could declare success, and I'm happy to talk about anything you wish, or I could continue to show slides, and that would probably take another 7 minutes. Okay. I'll tell this story backwards. I went to go visit Far West Fungi, which is down in Monterey, near Monterey, in the strawberry fields. Okay. I'll tell you why and how I got there in a minute. Some poor quality photographs off my iPhone. This is what it looks like. There's some sheds, there's some piles of stuff. The stuff turns out to be sawdust, which is free to get. Wood chips and sawdust are free to get, apparently. And they take that and they put it through an autoclave to sterilize it in little bags, which I'll show you in a minute. They add to it crushed oyster shell and some rice husk. It comes off this assembly line. Those bags are on the right. They've just been sterilized, and to this they add a slurry of a wood fungus, a solution of wood fungus to inoculate the bag. Okay, they then incubate that bag for a period of time, maybe 72 hours. You can see the fungus starting to grow, the white material in there. And over a 72-hour period, this block of material, mostly sawdust, will compile into a physical intact block. It won't just be dust, loose dust, it'll be solid. You take that and rack it, and what they're doing in this example is cutting the plastic wrapper, and then fruiting bodies come out, which look like this when you zoom in. They begin to look like things you would eat. Other types of cultivation and different types of fungus growing, and so on. And it ends up in boxes like this and goes to the supermarket, where it's really beautiful and tasty and is organic mushroom. So this was eye-opening to me at one level because it shows that food can grow from wood directly. And in my world, when people talk about biofuel manufacturing, they say, well, we're going to do cellulosic biofuels, so we're not competing directly with the food supply. We're not taking corn syrup into fuel. We're going right from the wood into fuel. And this sort of brought home that wood is food, right? These things are half protein and half carbohydrates in terms of if you look at the nutrient cut. And a really impressive operation, a family operation down there. Now, the reason I had gone there was this fellow named Phil Ross, who's an artist up in San Francisco, and this is an instance of the front page of his website. Trained in biology, coming from cooking, learning sterile technique in the kitchen, and then becoming interested in what you can explore and make with biology. And he's using these fungus to grow different things, not necessarily food. His website's just his name with no space, right? You can find it pretty fast. So for example, he took these bags and made a different type of brick out of a fungus associated with a tea, and in a provocative museum piece, assembled these bricks into a primitive tea house so people could go experience a tea house that they could drink directly. All right? But he's also growing other things, again, somewhat provocative by not being finished in their surfaces. By growing the fungus with this 72-hour compile time and then baking it, you get to something that's structurally pretty impressive. Now there's wood, obviously, but the seat and back and sides of this chair are from the sawdust input with the fungus compiling it, right, and then baking in, and there it is. There's a company now, Ecovative, if I remember correctly, which is growing fungal materials for packaging, inserts, and other types of manufacturing. This is pretty exciting for me, these fungal growth systems, because they let me imagine being able to compile macroscopic objects from microscopic things with an input that's an abundant input. Now, all of Phil's work, which is genius in my opinion, does the shaping via extrinsic control, via the mold that shapes the dust and what the fungus grows into. But what if we could take genetic programs and implement them within individual fungal cells, such that we could get to a complementary patterning approach, which would be bottom-up program pattern formation. This work here, borrowing from Radhika Nagpal and colleagues, working over a decade ago on what was then called amorphous computing. How do you program systems to grow into shapes? Right? Or to think about it more abstractly, what programming language would you use to control a pattern in 4 dimensions? Not just a finite state machine operating over time, but an ensemble of agents that aren't on a grid, aren't on a clock, and are using local information to make decisions and differentiate, and then nevertheless still grow into the shape you prescribed at the outset. So I rerun this movie from Radhika. It starts already a little bit into the movie, but what's happening here is a cell at the center of the movie is replicating. Its descendants are transmitting information locally, differentiating, and continuing to grow into the final pattern that you see at the end of the movie. This is a simulation. This is not a physical experiment. This is a computer simulation of an early amorphous computing program. And the simulation stops around here, with the color representing the state, right? And that's about it. Now, the language that Radhika and colleagues were playing with to implement this cross was just a colony growth sort of language. Replicate out to a particular radius of your colony until you hit the prescribed boundary, prescribed by the programmer, right? So the programmer at the outset of the experiment would trace the shape they want these cells to grow into. And then the compiler compiles down the instructions that get placed in the mother cell that initiates the whole system. And as those cells grow and divide, they're all running the same program, but they're differentiating state within the programs within the cells. And so you grow circles and keep growing circles until you fill in the external trace. Right. So this is a pretty old example, but a simple example of how you could think about implementing a language for controlling a system in 3 dimensions, x, y, and t. Right. And you could argue it's just 2 dimensions of x and y, and then it sits there. That'd be fine. So what if I start to implement, so I'll back up. The capacity to program and implement this really is in part limited by the amount of state you have that you can control, and also by your bandwidth for communication. Right? The rate at which you can get to a pattern depends on how much messaging you can pull off. And obviously you can imagine wanting to integrate signals through logic elements. Right? So if we can take our systems, which we've developed in bacteria, and move them over into this fungus, and scale them up a little bit, we have a chance of actually bringing to life the substrate to implement program pattern formation within the fungus in a microscopic bottom-up way, complementing what can be shaped with the extrinsic information. And that'd be a heroic bit of work, just to say it for what it is, and how I'm relating it might not be perfectly right, but that's how I'm thinking about it. Part of the reason I'm thinking about this is because throughout biology, we see a lot of different types of programmed pattern formation, which is an understatement. We're all familiar with this. I'll just give you a weird example coming from the world of sponges, where in the ocean, you see sponge pulling together silica and making pretty impressive stuff, which includes structural members, fiber optic cables, and so on. This is the basket sponge from the Pacific Ocean. And its skeleton shown here, work done very nicely by Joanna Eisenberg, then at Bell Labs, but now at Harvard. So what if I could implement program pattern formation in wood fungus? What if I could power them with sawdust? And what if I could begin to bring into that platform for replication and pattern formation biocatalysis of materials and deposition of materials? Right, so that's what I'm thinking about, sort of moving towards on a next decade timescale as a growth market, if you will, for logic memory communication. Here's another example of program pattern formation, which I just have found impossible to ignore now, being out here for a couple years. I have a pine tree like this in my front yard in Menlo Park, and right this time of year, it starts doing this. It starts making pine cones. And the pine cones are enormous, and they fall off in a month or so, and they get taken away, and we do nothing with them. So I'm very curious about this sort of surplus manufacturing capacity we have around here. In Menlo Park, if you look at the composting rates in Menlo Park, it's, so far as I can tell, about 16 million pounds of garden clippings a year, which is 500 pounds per person per year. That's a lot of matter being compiled annually and not being used for manufacturing. So it's a big picture dream, but I'm keen to take ownership in a limited sense of program pattern formation, couple that to inorganics and deposition of inorganics and change how we begin to manufacture stuff. So that's the fungus story for anybody who wanted to know what that was. And this might be my last slide.
Done. I mean, truly, people have been talking about circuits and genes for a while. This is really coming close to real logic. It's very impressive. But coming from the engineering profession, a lot of people who view this see people calling it engineering biology who aren't engineers. The engineers themselves go, well, this is basic science akin to when the Nobel Prize was awarded for developing the transistor. They got it for physics. And so the engineers tend to think we take pride in building things to solve a problem or serve a purpose. And I don't think it's there yet. So it seems a bit of a misnomer to call it engineering versus modification or something maybe less grand. And I don't mean to be argumentative. It's a philosophical point that engineers do bring up a lot in our hallway conversations. That is this really bioengineering or engineered biology? Isn't it really exploration in the sense of physics begetting the transistor, then the circuits follow?
It's a good comment and questions along with it. And I'll have to think about it, honestly. What I experience is bioengineering, whatever it is today, exists in the context of everything that's come before. And so we are burdened with and inspired by simultaneously the work to develop the hunks of metal that were reference objects for measuring current in wires and improving manufacturing and telegraph engineering in the 1800s in Britain. The basic science leading to or applied science leading to the transistor, or whatever it was. Meade Conway, VLSI, decoupling design in fab. William Sellers and standardization of screw threads in 1864 in Philadelphia. The work in Segovia, Spain 2,000 years ago to standardize rock. The work in Stanford over 100 years ago to grind up artificial stone and make that fancy tiled diamond pattern around the original quad. All of this hits us simultaneously. And so it's an overload to experience it as somebody working and trying to figure out where is my valid home. I'll simply represent that we're doing the things we're doing. I feel that I'm an engineer. I'm trained as a civil engineer. I worked for Amtrak one summer on construction management projects, and I'm shocked come into the world of biology and not see the engineering infrastructure well developed. Whether that's because the science isn't there or the infrastructure isn't there or both, I don't know. Stanford having helped invent genetic engineering a human generation ago, never started a department of genetic engineering. Here we are. Other things I could represent over the last year would, for example, highlight papers where if you take the idea of a standard biological part, we've now moved that area of work forward such that people should ask, don't just tell me what the part does, but tell me how likely the part is to do that function as it's reused with other parts, as the environment in which it operates changes. In other words, a quality score, a second-order statistic. And we proposed doing that work in 1999. It took us 10 years to get the funding to do that work because all the funders just wanted us to cure a disease. They didn't want us to make all combinations of parts and see what happens and then develop a mathematical model to interpret variation across changes in context. But having done that work and scoring various types of junctions among parts that were really poorly behaved, meaning high variation, we could then prioritize a next round of work to see if we could push that variation down, and we did. And nobody had done that before. And the answers oftentimes are in the natural genetic architectures that we don't know how to appreciate, but when we bring first-order engineering goals to the biological substrate, sometimes we see the biology in a new light and get the functional compositional solutions we want. And then sometimes in the example I showed here, we come up with new architectures that we don't see in the natural examples. So I don't honestly know how to directly respond to your question, and I'll just simply relate we're at an interesting time where every past experience is sort of converging on biology as a substrate, as a type of material. And so what do you make of that? if I were to pause, zoom out, and come back into the question and mount an argument in favor of bioengineering as an engineering discipline, I would immediately represent that we don't have the discipline right now. But I would also argue that we could expect the discipline to develop in a way that's likely to be sufficiently unique and independent of other modes of engineering. And mostly this comes to the physics of the substrate. Biological material is a different type of material. We've got self-mixing systems due to the thermal noise. We have very high heterogeneity within the systems. It's not like just repeating the same thing over and over again. So it really pushes beyond any existing tool for complexity management I've seen. We've got reproducing systems. So how do I design a system that exists not just on the timescale of performance and learning via memory, state change, but it exists on this third time scale of reproduction. And then it also has this, as a comment as somebody walked in looking at the title slide, I think said, oh, that looks dangerous. If we actually make any progress on getting better at engineering biology, everybody freaks out. The ramifications on this area of work are just impressive. And there's one more reason that doesn't come to me right now, but I would argue that it's already apparent that the living matter is just a different type of stuff, and the track record over the last decade is we're actually getting systematically better going around the design, build, test loop, and if we keep pushing on that for a long while, I think you get to a new engineering discipline that's not just on a PowerPoint slide, but actually is substrate distinctive, and thus engineering solution distinctive or engineering approach distinctive. So I'd value talking about it more, right? It was just those sort of two things would be the major modes of what I'm reflecting.
Engineers, it's physics till you ship a product. That's the mantra.
Yeah. So genetic engineering, how much money did electrical engineers ship in terms of product? What's the market value of electrical engineers' products in the U.S. last year?
Globally, it's about 300 billion, electronics industry.
So in the United States, genetic engineering products were 300 billion last year.
Oh, no, I've heard Rob Carlson's talks too. I mean, this is...
Right. No, but I think there's that element too, which is these tools are being used to ship product, right? The market for genetically encoded data storage that operates in live cells today is approximately $0 a year. And the product we're shipping, our logic gates are being immediately reused by people in the medical school who are studying how bacteria colonize the gut. And they want to know what the signals are at different crypts in your gut, right? So they're research tools, I would say. But there's GenEncore with 1980s engineered subtle lysine for better soap, right? And that has a market impact, and it also has a impact in reducing our domestic hot water energy requirements, so you don't have to heat up your wash water temperature. I'm sorry?
Disulfide versus.
Yeah. So I would take issue with we're not shipping product. That's wrong.
this stuff. Ship product from what you're talking about here. That's what I mean. Sure, people are doing genetic engineering and shipping slightly modified subtolysine by the kiloton. There's no doubt.
Yeah.
But no one's shipping bacterial logic.
I would agree. And we didn't have it till this month.
Very good.
Yeah. But yeah, I would agree. That's a good point. And we certainly have to do that, right? I mean, if we don't develop markets and products around this, it doesn't go, period. I don't see how it goes.
Chemical that changes state?
In the test experiments, it's an exogenous factor. In the applications, like what I just mentioned, where people are using it to assay and record state within a mouse gut, it's endogenous transcription factors. So you could imagine a natural microbe. It sort of goes back to the gentleman's question earlier. You can integrate. The neat thing about these logic elements is they take a transcription current in, they take a transcription current out. They're a pretty compact package. So you can insert them anywhere in a natural chromosome where there's a transcriptional current, right? And so in the application sector, when you get to applications, you're driving input, output, and control signals with the native signals. You're basically patching in to the native transcription signals. Does that make sense?
Well, changing state.
Yes.
You just add a, I mean, you have an environment, you just add a.
Yeah. So we're doing switching here. We have cells, they have the switch running, we're adding a sugar or a surrogate of a sugar, and the cells respond to that. They initiate a transcription signal that makes the enzyme that causes the thing to switch. All right, so we have to add a molecule to the culture bacteria that then gets transduced into the cell to create a transcription signal that triggers switching. I have a pod of bacteria, I add a sugar to it, the gates switch. Yeah. If I wanted to apply them, so that's just for developing them, testing them. So we can control the adding of the sugar. When you go to apply them, you want to connect into the natural signals within the cell, or whatever it is you're trying to react to. So you integrate the gates into that context. In these experiments, certainly. Right? These are the best studied switching sugars around in biology. Arabinose, right? Anyway, yes. But they're just the experimental tools, right? It'd be like asking, is that a special switch, right, from Texas Instruments? Yeah, it's special, right? But it's the one that works. Yeah.
Right. After it switch, if you want it to switch back, then what should we do? The sugar
Okay.
is already.
For the architectures I'm showing here, these are permanent gates. They're write once gates. So they're only switching and they're not going back. Okay? If you want to see if you pull the paper that's referenced in the talk and you go into the supplement, figure S2, supplementary figure 2, here's the architecture for the rewritable gates. Okay? And you just basically have to reverse a lot of the architecture of the switches. So instead of pulsing the integrase, we're going to pulse the directionality factor. Right? So you just reverse things up a little bit, and this will push and then revert back spontaneously. Right? So just check out supplementary figure 2, see if you can make sense of it. But pretty simple changes get you rewritable. And this is what the gates look like for rewritable. Yeah, excuse me. You could make rewritable gates that have internal state or memory, right? Like with the data register, but that would be more complicated, right? So what I'm showing you here is sort of a push and relax architecture. So you might just have to play around with what these symbols mean and check it out, or talk with me later. Yeah.
The applications that you mentioned towards the beginning of the talk had to do with something like, say, a counter for biological events.
Yeah.
Now, before such a counter becomes practical, how much of a speed up do you think we would need in the switching speed of these biological circuits before we could do something?
I think we got to get to 15 minutes, right? So I think we're going to be at the performance limits of the enzymes, right? So that would be an 8-fold improvement over what we shipped in May of 2012.
What's your doubling rate?
Yeah, but so the fastest doubling rate you're going to get for E. coli.
What was your time? The last time you measured before that, you said you were 8 minutes.
Oh, well, we went from what did I say, 10 hours, 12 hours to 2.5 hours.
So it's.
Right.
Better than
But you know, let's not go there, because I don't have decades of data to show that trend. Right.
Moore's Law, anyone?
Yeah. Yeah.
How much work do you estimate it would take, or do you think it's feasible to do that, get it down to 15 minutes?
I certainly think it's feasible. Couple months. That's what your project focus was? I mean, our focus is on exploring different logic element architectures. Jerome's super excited to ship a product, right? He's going into medical diagnostics. You know, he's got these gates being driven off of urine samples, right? So this is not a populated space. There aren't a lot of people working on this, right? Tim Liu's group at MIT has done some nice work in this space, and I think so. I mean, people are starting to use these pretty fast. But very few people are working on this. The number of biologists working on serine recombinases of the type we like most of all are 2. Graham Hatfull in Pittsburgh, are you working on them? No. And Maggie Smith in England? Right, so this is not a hyper-populated space. Yeah.
What is the patent situation in this space?
We pushed our logic element architectures into public domain, care of Stanford paragraph 3 patent policy. We filed on the data register because others had filed out of BU and MIT, and those filings looked, well, I won't say more. But we think we're far enough ahead and have priority around the logic elements that we just pushed it public domain.
The world changed. First to invent is first to file.
Yeah, but we did this last year.
Okay.
and have a lot of things documented through DOI and DSpace and stuff like that. So I like our chances on the logic stuff. Yeah.
File, so yeah.
There's a lot of, but typically this space defaults to patents and uses of genetic functions, right? So the Supreme Court decisions that are in the US playing out right now, the Bowman versus Monsanto case having to do with whether or not patent exhaustion applies as product replicates itself. That would have some impact on this space. The Myriad genetics case around the breast cancer diagnostics, which is probably thought of as a more important decision, has more to do with composition of matter, not uses of function. So that actually won't impact the type of work I do. So I would say the default path is a lot of patenting and messy patenting and more patenting than commercialization, if you will. So I don't think that's a good balance. And puts a lot of things at risk in terms of integration, especially as the design build test cycle changes. We're actually reusing parts now. We're actually abstracting things. We're actually outsourcing compile and separating design and fab. And we gotta make some architectural improvements around property rights. For example, I don't know if anybody here would get up and cheer for the DMCA, but one of the nice things about that act is the carrier exemption, such that if you're intermediary redistributing copyrightable works, you're not accruing liability associated with that, so long as you take to a first approximation as I understand it. But in the world of technology where patents are used, you don't have the equivalent of the carrier exemption, right? So if I'm a DNA synthesis company, if I'm a gene synthesis company, if I'm a plasmid redistribution house shipping around DNA molecules, I'm actually accruing liability by enabling people to infringe potentially on third-party patents, and I have no way to clear that. Right, I have no immunity from it and an exemption from it, and no way to clear FTO. And that problem is, for example, disallowing people in companies who build automated strain engineering, new fabs basically that are better than anything else that exists. It's not allowing them to open up to outside users. We can't implement MOSIS in partnership with academia and industry in genetic engineering right now because of the property rights puzzles around liability associated with intermediaries handling patented materials. So from the microscopic, there's filings around this type of stuff, and then at the macroscopic, the architecture isn't working very well. Another way to think about it is biotechnology is going to struggle to figure out, is it going to reorient itself from sort of deep verticals focused on specific applications to many, many more sort of layers of platform tech. And if it does that reorientation, the property right's going to have a lot to say about it.
Looked at or tried using the biochemical changes that happen in the cell during division, or just like right up into division, as the input for flipping the switch?
Yeah, that's the thought. I mean, to be quite honest, we're close to having a push-on, push-off toggle. right? And that's been most of the focus on that over the year. We have shipped a bunch of theoretical designs and analysis of how to scale up counters to 8-bit combinatorial asynchronous counters and other types of architectures. Those are all theoretical, and we're limited with 4 integrations right now. So practically, we haven't pushed too hard on integration into the application. We know that there are cell division cycle dependent signals. It'd be interesting to know whether or not a lot of work to do, right? So it'd be interesting to know, should you plug into a transcriptional activation signal, or should you plug into a proteolysis signal? There'd be different types of signals. But it's a little bit too early for us. We don't have a systematic program pushing towards that right now. Another way to say it, just to give you a little bit of context, what I'm showing here represents sort of the end of a 10-year, 11-year research cycle. Right? Everybody associated with these projects is graduating with degrees or postdocs and getting other positions. And so we're really in this period of renewal. And it's why you hear me explore something seemingly wacky like wood fungus, and what could we do with that? Well, maybe that's what we're going to do, or maybe we return and say, actually, we're going to get an 8-bit counter that works in stem cells, human stem cells. Let's go do that. And there's no chance that we're at risk of saturating the space, either from research or application. So we know of those things, they're peripheral in the adjacent, and we'll see what we or other people do. I'm sort of hopeful, perhaps not completely naively, that because we've taken such care to implement our systems in ways that are agnostic to the inputs and the outputs, that it'll be easier for other people to reuse them. And we're starting to see that. Not with the data storage so much as with the logic elements. There seems to be a lot of demand in the research community to use those. I don't know if that counts as shipping a product. It doesn't sound like soul of a new machine quite yet. But we'll see. NSF. DARPA has a Living Foundries program, which is working on tool layer stuff. The biggest public funding's overseas right now. China and the UK have the most focused programs to do work like this. Minister Willits in the United Kingdom is a significant champion of getting better at engineering biology. But in the US, I would say NSF and ARPA are the folks who are investing in the get better at engineering part, and nothing else is super well organized. I would acknowledge that a significant development has been NIST out of the Department of Commerce in the US. So National Institute of Standards and Tech is making synthetic biology a priority area for investment, and they're shipping people to Stanford, who just started this week. They're shipping a group of people to Stanford to work on biological measurements led by Mark Sallet. So he just sold his house in Maryland and bought a house out here, and he's coming out here to lead a group to work on stuff. And metrology work is going to be a big deal. We have no reference objects that work inside living cells to first approximation. And as we've explored that just a little bit, they work really well to remove types of variation and make measurements reusable across locations and over time. So it's kind of shocking that basic work hasn't been done in support of engineering. So let me acknowledge NSF and ARPA and NIST newly to the game. DOE is obviously spending a lot of money, but they're spending that money on sort of targeted projects, making bulk commodity products like biofuels, and less on infrastructure. Although, to be fair, they're spending a lot on scientific infrastructure, like the Genome Center in Walnut Creek that sequences microbes in plants. So I want to give them credit. But the Obama White House released the bioeconomy blueprint in May, about a year ago, and it hasn't been really popular to talk about it politically. There is not a plan for advancing biotechnology. There are not things that lead to structured public-private partnerships. There are not industry consortia that say, wow, this is strategically important for us to get a lot better at building silicon or whatever, building DNA, right? Just none of that maturity is in the space. So I shouldn't represent that we have a plan. We neither have a plan with respect to tech development. We also don't have a plan with respect to policy. Right? So, and people are really struggling to understand at a policy level, what is the significance of decoupling design and fab? What is the significance of having standards that enable coordination of labor and reuse of parts? All of our frameworks, for example, for biosafety are coupled to a strong institutional oversight model, where Stanford's in charge of biosafety at Stanford, and so the institution will take care of it. And the people at Stanford will be responsible to the institution, responsible to the government. But if the recent example on Kickstarter of a group raising so far maybe $250,000 to make a glowing plant, right? Outside of a strong institutional oversight framework. Who's in charge of that? Who's responsible for that? Well, right now that's defaulting to the civil society organizations that are against promulgation of genetically engineered organisms. Right? So they're trying to run a Kickstopper campaign to counter the Kickstarter campaign. So that's the state of play, right? Just sort of expanding from your innocent question, who's paying for all this? Yeah, it's not very many people, and it's not well organized, which creates a neat opportunity because it's not that expensive either. The team I work with here is 5 people. Right? Yeah.
What are the safety issues here, short-term and long-term?
The meta safety issue is we're doing okay with biosafety. It's not perfect. We had a 25-year-old researcher die in San Francisco last year from the VA. He was working with a natural microbe associated with meningitis, if I remember correctly. He infected himself on a Friday and was dead Saturday in his Treasure Island apartment. So laboratory biosafety is a real issue. That issue predates genetic engineering. Paul Berg and others led the conversation around how to wrap genetic engineering into the biosafety framework, and that's worked pretty well. It's not perfect, but it's worked pretty well. You could almost say it's worked too well, because we spend too much energy on it, we don't take enough risks. So there's another type of death, which is associated with lack of progress. But the meta issue with respect to biosafety is generational renewal. Right? So my students weren't alive when genetic engineering was invented. And so they'll grow up in a few years and be the leaders with respect to biosafety, not just the practitioners. And so we sort of have to pass the baton of critical thinking and community leadership and responsibility to a next generation of people. And that's possible to do. Security is a different topic. So how do you deal with intentional misapplication of biotechnology? Nobody has a plan for that. There's a very nice course being offered at Stanford right now called Surgery 222, offered by an ER doctor, and the topic is biosecurity, and she's done a brilliant job of bringing people together to talk about it, and the students in that course are amazing. But I would be fair to say nobody has a strategy for securing an open distributed network that's based on the living world. And as people can increasingly inject intention to that. I don't consider that to be an immediate short-term risk, although bad things could happen right away. I think it's a longer-term strategic puzzle that's probably one of the bigger ones around. Yeah.
Potentially long-term for the artificially intelligent growing crud that takes over everything.
Well, it already has, right? That's the beauty of this, right? Biology's already taken over the planet.
Yes, that's right.
Right.
Yes.
So as George Whitesides, observed, green. You don't have to worry about gray goo. It's green goo. It's already won. And
Green goo.
what's that?
Like a beautiful green goo.
Yeah.
It's running this time.
Yeah. You need to be careful, right? Because if they're 10 to the 31 phage, you can infer that there's several orders of magnitude more bases of DNA, right? So like 10 to the 35th, 10 to the 36th bases of DNA on the planet. We've sequenced as a civilization 10 to the 15th today, if I believe Huanming Yang's numbers from the Beijing Genomics Institute. So we've got 10 to the 20-fold to go, just in terms of sequencing, if we could somehow magically sequence every base of DNA on the planet, including many redundant bases. Our synthesis capacities aren't anywhere near that. They lag sequencing. So yeah, you could release things at 8 amplify and take off, but in a way that'd be a success of evolution, not engineering. I would agree with that scientific versus engineering frame. And I don't think we're going to be able to do that, to be honest, anytime soon. I think the limited environments that we impact directly or indirectly, which are big environments, is where things will play out. And just to put a number to that, right, we do impact something like 75% of the land, arable land on the planet. So that we're impacting a lot already. So I don't want to say it's not going to be a lot, but in terms of nature, natural biodiversity, I don't think we're going to have a big impact, except the impact associated with what's going on anyway, which is horrible loss of diversity through ecological change and destruction.